mitochondrial genome. First structure of mitochondria, its outer membrane, inner membrane, its double membrane, curved organelle, and in inner membrane, these curves are called cristae, and the area bound by cristae is called matrix, and it have ribosome and mitochondrial DNA. Apart from nucleus, mitochondria also contain DNA. How scientists are viewing this aspect as like there are two theories related to the origin of mitochondria. One is endosymbiotic origin and second is autogenous origin. First, endosymbiotic origin. This is proposed by Andreas Kimper in 1883, but extended by Lynn Margulis in 1980s. According to this origin, what this is that mitochondria is once a free living prokaryotic cell which can capable of implementing oxidative mechanism. Then uh, a, a eukaryotic organism engulf the mitochondria. Then the mitochondria start inside that eukaryotic organism. Then they started living in symbiotic relationship. After that, it started to like that mitochondria started from like passing from one generation to another. This is like how they are viewing this aspect. Then second one is autogenous origin. In autogenous origin, mitochondria were born by splitting the portion of DNA from the nucleus of the eukaryote at the time of divergence from prokaryotic cell. This is autogenous origin. But as mitochondria is having more similarity with the prokaryotic cells like uh, this like ribosome is 70S, unless in eukaryote it's 80S, then this cristae inner membrane is similar to that of prokaryotic organism. Then this is circular DNA, which is also similar to that of prokaryotic organism. So this is like endosymbiotic origin is widely accepted than autogenous origin. Next, mitochondrial genomes. The genome consists of circular chromosome that is 16,569 base pair in size. It's located in mitochondrial matrix. Consists of two strands, light strand and heavy strand. This region is called D loop. That is 1121 base pair long. And this is the site where replication and transcription is mostly controlled. And it consists of 37 genes, as you can see in the image. That 22 tRNAs are shown by dots, and 2 rRNA and 13 proteins are like the outer shaded region. Those depicts that 2 rRNAs and 13 proteins. And these 13 proteins are subunits of enzymes of oxidative phosphorylation. Another 74 comes from nuclear genome. And this like this shows the size of mitochondrial genomes in different species. And this slide shows plant mitochondrial genome sizes. Now let's see the difference between mitochondrial DNA versus nuclear DNA. First, like mitochondrial DNA contribute only 1% of the total DNA of the cell. 99% contribute by nuclear DNA. Nuclear DNA found in nucleus of the cell, and this is found in mitochondria. Two sets of 23 chromosomes, and mitochondrial DNA have several copies of single mitochondrial DNA molecule. And nuclear DNA comes from both maternal and paternal, but the mitochondrial DNA come from maternal only. Then double helix circular bounded by nuclear envelope. This is free of nuclear envelope. And now mitochondrial DNA replication. First, the supercoil circular mitochondrial DNA uncoils. Then now like heavy strand starts to form the displacement D loop. Then loop expands. Then the D loop looks like in D shape, and L strand starts to form. And after that, replication complete, and two circular mitochondrial DNA supercoil are formed. Then transcription, both strands are transcribed. The D loop contain like one promoter for each strand, and the entire strand is transcribed. Then the RNA is cut into individual RNA for each gene. Then protein coding genes are given poly A tails. This is like something which increase the stability of RNA. And rRNA and tRNA molecules are modified. And translation, mitochondrial RNA have no 5' gap. So like FMET tRNA, that's the thing which initiates protein synthesis. And mitochondrial mutation, like mutation is very high as compared to nucleus, that is like 10 to 17 fold times higher. Then why it is this much higher is like there is repair mechanism, but that is not that sufficient to counteract the oxidative damage. So like then protective histones are also lacking. Then and this rate mutation rate can be also increased by environmental agents or by like mutation in nuclear genomes which involved in maintenance like different processes in mitochondria. Then three types of three types of mutations are seen in mitochondria. One is missense mutation in the coding regions of genes that alter the activity of an oxidative phosphorylation protein. Uh, a missense mutation is a mistake in the DNA which results in wrong amino acid being incorporated into a protein because of change. The singular DNA sequence change because of that which amino acid, I mean, which like codes for different amino acids. Then point mutation in DNA RNA that impair mitochondrial protein synthesis. And third is deletion or duplication of the mitochondrial DNA molecule. This is somatic in origin and only small amount is inherited in some diseases. Then let's see about mutation in mitochondria and disease. Like in US, like according to some about 1 in 4,000 children in US develop mitochondrial disease by the age of 10 years. Like more than 100 different rearrangements and 100 different point mutations have been identified in mitochondrial DNA that can cause human disease, often involving central nervous system and musculoskeletal systems. The disease that result from mutations show different patterns in inheritance because of three distinct features of mitochondria. One is replicative segregation, homoplasmy, and heteroplasmy. Then another is maternal inheritance. First is like replicative. First, let's see about replicative segregation. It's like a cell division, multiple copies of mitochondrial in each of the mitochondria in a cell replicate and sort randomly into a new cell. That is called as replicative segregation. And homoplasmy and heteroplasmy is like homoplasmy can be seen like pure population of normal mitochondria, that is without mutant. And like heteroplasmy is like it contains mutant population too. Then maternal inheritance it inherit from only from the mother side. And as you can see here, that first it's like 20% mutant. It can contribute to different percentage of mutant. 20% it's like no problem. Then 50%, mild disease maybe, and 80% it may cause severe disease. And it's end of this.